All right, this is part one of meiosis and sexual life cycles. This is really the beginning of our study of genetics, which is the next chapter. Genetics is the study of heredity and how traits are passed from generation to generation. We've already talked a bit about variation between individuals and how individuals reproduce using sexual reproduction is a big part of that variation. What's actually being inherited from generation to generation is not the traits, but the genes and the variations in those genes that we call alleles, and we'll hear that word more in the next chapter. But it's not the traits themselves, but the genes that control the traits. And meiosis is, in the formation of gametes, eggs and sperm, is step one of that variation being passed on to the next generation. So genes are really the units of heredity, and they are found on chromosomes. And so some of the things that we'll cover in this section are exactly how these chromosomes are partitioned into the gametes for the next generation. And each individual chromosome has certain genes that are on that chromosome. And unless there's some kind of a chromosomal rearrangement, that's where we will find them in that particular species. And we call that location a locus, which is just Latin for location. Many organisms do not use sexual reproduction and also don't have meiosis, or they have reproduction. They have both kinds of reproduction, asexual and sexual. So when reproduction just uses mitosis, the kind of division we talked about in the last chapter, then we call that asexual reproduction. That prefix A there meaning not sexual reproduction. So in this case, mitosis is the type of cell division, and remember, in mitosis, the daughter cells are identical. The number of chromosomes doesn't change. Every round of division, you get two daughter cells that are identical to each other and identical to the original mother cell that divided. So in this case, in asexual reproduction, all the offspring are going to be identical genetic clones of the one parent. And there are organisms that have both asexual and sexual reproduction. Some only have asexual, like amoeba that we looked at in lab. Amoeba only divide by mitosis. They don't have meiosis or sexual reproduction. But there are others like the paramecium here that we also looked at. Paramecium can do both. They can divide by mitosis, and in that case, the daughter cells are genetically identical. And they can also go through meiosis and fuse with other cells then to form temporary diploid cells that are going to go through meiosis. Plants can have both sexual and asexual reproduction. If you use cuttings to reproduce a plant, or like if, for the example, strawberry plants and other plants send out runners that then sprout into a new plant, all those daughter plants that are going to be produced by the runners are all going to be genetically identical. Trees often do this. Root shoots will come up and develop into a new tree that's going to be genetically identical to the tree that the root shoot originally came from. That's asexual reproduction. Whole forests are produced that way. But plants also have sexual reproduction, almost all plants. Like, for example, strawberries, the seeds on a strawberry, seeds are produced by sexual reproduction, and part of that is meiosis, the other kind of cell division that we're going to talk about in this chapter. There are even some animals that have both asexual and sexual reproduction. This is a hydra. They are in the same group of organisms as jellyfish, anemones, corals. It's sort of like an upside down jellyfish. They have stinging tentacles. They do produce eggs and sperm and can reproduce by sexual reproduction, but they also can reproduce by budding. And there are some types of animals that can reproduce this way, not mammals. If we could reproduce this way, that would be, like if a lump formed on your side and then it got bigger and bigger, and then eventually a toddler fell off, and the toddler would be an identical clone of you. That would be asexual reproduction. Mammals don't have asexual reproduction. But some animals do. So if it's produced by asexual reproduction, that means it's a genetic clone. It's identical. Here's an example from a forest. So here's a dead tree in the center here that you can see, and around it is a circle of trees that have grown up as root shoots from that original tree. So these trees are all identical genetic clones of the original tree here that died. Sexual reproduction, on the other hand, we have two parents, not one parent, and the offspring are going to have about half of the genes from each parent. So every generation you're creating recombination. Each individual is going to be genetically unique. We're going to use a type of division called meiosis to produce the gametes. For animals, gametes are eggs and sperm. And this type of cell division is only found in eukaryotes. So prokaryotes like bacteria and archaea do not have sexual reproduction. They don't have meiosis. They only divide by binary fission. They do have ways of exchanging their DNA, but it's not sexual reproduction. It's unfortunate for students of biology that meiosis and mitosis are so similar words. They both start with an M, they both end with an SIS, so how to remember which is which. So I will give you a hint for how to remember that meiosis is for producing gametes, like eggs and sperm. If you look, you can see that there's an egg and a sperm right in the word. So the O there is the egg, and the little E here sort of looks like a sperm with a curled tail. So the one that has an egg and a sperm right in the word, that's the one that's for producing gametes. During meiosis, the whole point of meiosis is to reduce the chromosome number by half so that when fertilization occurs, the normal chromosome number is restored. If meiosis did not occur, the number of chromosomes would double every generation if fertilization occurred. So gametes generally have half the number of chromosomes as the parent, so human eggs and sperm have half the number of chromosomes, and almost all human adults have 46 chromosomes, and so gametes have 23 chromosomes. Meiosis has 2 rounds of division, not just one, and so we're going to end up with 4 daughter cells that are all genetically different. Each one is genetically different, unlike mitosis, where we had just 1 round of division, 2 daughter cells that are genetically identical to each other and to the original mother cell. In meiosis, the 4 daughter cells are all genetically different from each other. And they're different from the mother cell as well because they have half the number of chromosomes. Chromosome number for each species is unique. Although there are vastly different species that just happen to have the same chromosome number, they wouldn't have the same genes or the same versions of the genes on there. But we need some words to talk about chromosome number. So for animals and most plants, we are diploid species, and diplo is the Greek root for double, so that means we have 2 of each chromosome. So our 46 chromosomes are 23 pairs of chromosomes, which you've probably heard that before. Haplo is a root that means single. It's kind of tempting to think it means half, but it means single. So a haploid cell or a haploid organism has only 1 copy of each chromosome. And the number of unique chromosomes in an organism we call the N number. So N for humans is 23 because humans have 23 unique chromosomes. And so our diploid number is 2N. So if the N number is 23, the diploid number is 46. So just knowing the number of chromosomes for a species doesn't tell us whether it's diploid or haploid. So if we want to know that, then we ask, what's the ploidy of this cell, or what's the ploidy of this organism? And what we want to know isn't how many chromosomes it has, because if I have a cell and I say, what's the ploidy of this cell? And you say, it has 4 chromosomes. That doesn't tell me if it's diploid with 2 pairs or if it's haploid with 4 single chromosomes. So the answer to the question, what is the ploidy, is it's diploid, it's haploid, or you can use N. It's 2N. We do have organisms that have more than 2 of each chromosome. So tetraploid would be 4, octaploid would be 8, dodecaploid is either 12 or 20, I forget. Aneuploid is abnormal. Aneuploid is an abnormal number of, so you can see the root in here is actually an, meaning not, and u for good. So aneuploid means not good. It's a not good number of chromosomes. So aneuploid is abnormal. It's an abnormal number of chromosomes for that particular species. And we'll talk about, when we get to genetics, we will talk about some human genetic disorders that are aneuploidies. So here's human karyotype again. It's the same one that I showed you in the previous chapter. And we can see the pairs of chromosomes. And again, they're numbered based on their size. So chromosome number 1 is the largest chromosome, and everybody has 2 of those. All the way down to chromosome 22, which is the smallest chromosome except for the Y. And everybody has pairs of the 22 autosomal chromosomes. So let me actually put that on here, because I don't know if I have that. Let's see here. So the 22 chromosomes are the autosomal chromosomes, and then we have a pair of, that's not going to work, and then we have a pair of sex chromosomes. So the X and the Y chromosomes are called the sex chromosomes, and for the vast majority of individuals, if you have XY chromosomes, you're going to be male. If you have XX chromosomes, you're going to be female. Mostly, because nothing is absolutely the case for anything in biology. There's always exceptions to everything. Whatever general rule you make in biology, there's always, well, there's these things over here that don't follow that rule. And we have that for the sex chromosomes as well. And in general, the larger chromosomes have more genes on them. We have about 25,000 genes, and they're distributed on all of the chromosomes. The smaller chromosomes have fewer genes than the larger chromosomes, but surprisingly, some of the larger chromosomes have fewer genes than you might expect, just based on their size. So genes aren't absolutely equally distributed on the chromosomes. And what we can see here in our pair of replicated chromosomes here, we can see that there are 2 sister chromatids. So these are from a cell that is ready to go through mitosis generally. When we develop these karyotypes, they're from fully condensed metaphase or late prophase chromosomes. And so we can see the 2 identical sister chromatids, which are smashed up against each other and attached for the entire length. And then the reason there's 2, these are what we call homologous chromosomes. So homologous means they're not identical. They have the same genes in the same location, mostly, but they might have different versions of that gene because they're not made by DNA replication. And one of the things that we'll talk about in this chapter is the fact that your pairs of chromosomes, one of those came from your mother and one of those came from your father. So for your chromosome number 1s, one is what we call the maternal copy, it's from your mother, and the other is the paternal copy from your father. And so you have at least 2 copies of every gene, one on each pair of the chromosome. So here's a pair of homologous chromosomes with the sister chromatids kind of blown out here so we can look at them more closely. And what these letters represent are different versions of a gene, which we call alleles. So alleles are the same gene, but a different version of that gene. Like you might have one allele for blue eyes and one allele for brown eyes. Same gene, different version of that gene. So sister chromatids, our identical sister chromatids, are always going to have the same allele. So the capital and lowercase versions of these letters represent different versions of that gene. So the A gene, the B gene, and the D gene here. So the sister chromatids are always going to be identical. But if we compare our homologous chromosomes, they are not always going to be identical. So for the first gene, for the A gene here, they do have the same allele. But for the B gene, this chromosome has the dominant version of that allele, and this chromosome has the recessive version of that allele. Just generally, we represent dominant alleles with capital and recessive ones with the lowercase, although, as we'll see, that's not always the case either. So gametes are generally haploid, and for humans, that's the case. We are a diploid species. And so that means that the gametes are going to have half the diploid number. So for this species, whatever it is, where we have 6 chromosomes, that means we have 3 pairs, so the sperm and the egg are each going to have 3. And then when they fuse, the fertilization event, we call this initial cell that's formed a zygote. We don't call it an embryo yet. It's not an embryo until it divides the first time. When it hasn't divided yet, this fertilized egg is called a zygote. And it's, for humans and most other animals, that cell is going to be a diploid cell. Now, there are other types of union of gametes. Not all gametes have are a big gamete and a small gamete. Plants have a big gamete and a small gamete, although we don't call them eggs and sperm. We call them, generally, their pollen and ovules. But there are other organisms like most protists and fungi where the gametes are the same size. There isn't an egg and a sperm. There are just 2 gametes that have different cell types, and 2 different cell types will fuse together and still form a zygote. So we need some words for our different cell types as well. Somatic cells are just your ordinary body cells. So soma, we can see the root in here. Soma is Latin for body. So somatic cells, those are your regular body cells. Liver cells, brain cells, skin cells. These cells have 0 chance of contributing directly to the next generation. They are never going to form eggs and sperm. They cannot go through meiosis. Those are your somatic cells. The germ-line cells are the cells that might contribute to the next generation. Germ, in this case, we normally, in everyday speech, we use the word germ to mean something bad like a bacteria or a virus. But really, germ-line in biology, germ is from the same root as germinate or grow. So germ-line cells are the reproductive cells. These are the cells that are going to lead to eggs or sperm in animals or other reproductive cells in other kinds of organisms. So in animals, the germ-line cells are set aside very early during embryonic development. We can see these this is from a fetal male mouse, and this is the developing testes here. These little tubules here will eventually be the seminiferous tubules of the testes. Well, not in this mouse because he was sectioned up. But we can see these big fat brown cells here. Those are the primordial germ cells. Those are the only cells that are going to eventually divide by meiosis and make sperm. And similarly, in the female mouse, very early in the development of the female mouse, these primordial germ cells also are set aside. And especially in the female line, these cells divide very few times. Now, can you think of any advantage to that? That has been selected for by natural selection. Natural selection has selected for the fewest divisions possible. Germ-line cells set aside very early in embryonic development, and especially in the female line, very, very few cell divisions each generation. Why would that be an advantage? Why would that be selected for by natural selection? The reason is fewer mutations. So every time a cell divides, the DNA has to replicate and mutations accumulate. You have mutations that you did not inherit from your parents, mutations that occurred during the development of the eggs and sperm, or the egg and the sperm that made you. So all of us have a few new mutations. So minimizing the number of replications every generation is a way of minimizing the number of potentially harmful mutations every generation. So a gamete is the more general word in animals. Gametes are eggs and sperm, and all animals produce eggs and sperm. Even the hydra that I showed you, and lobsters and sea sponges, they all produce eggs and sperm. They're all animals. But in other organisms, they're the same size. And then we just call them, like in fungi, we call them plus and minus, or A and alpha. Some fungi have up to 4 different types of gametes. And then 2 gametes fuse. And the product of gamete fusion, or syngamy, is a zygote. And this is a special word that we reserve for that cell that forms from the fusion of 2 gametes. And it may be an organism that the zygote goes on to form an embryo, like in a human or other animal. And the zygote may go straight into meiosis and form more haploid cells, depending on the organism that we're talking about. We're most familiar with the animal life cycle because that's our life cycle. So in the animal life cycle, the entire life cycle is as a diploid, or sometimes tetraploid, octoploid organism. And the only haploid cells in the animal life cycle are the gametes. So the gametes are not capable of going through mitosis. If they do not fuse with another gamete, they are going to die. So for humans and all other animals, we have haploid gametes, mostly, unless they're a tetraploid or octoploid organism. And then we have diploid from the zygote to the adult. For the entire rest of the life cycle, they are diploid. So you can see the N versus 2N here. So 2N is diploid. Meiosis occurs in the adult and produces the haploid gametes. The haploid gametes can't do anything else. Now, most protists have kind of the opposite life cycle of that, and some fungi. And that is that they spend most of their time as a haploid individual, and the normal active individual for that particular species is the haploid individual. And they generally only go through sexual reproduction at particular times, maybe seasonally, maybe when the days start to get shorter, the pond starts to dry up, or other environmental cues could trigger them that it's time for sexual reproduction. And they're already haploid, so they can just turn into a gamete, and then they fuse with an opposite type of gamete, and this is where we would call them like plus and minus, or A and alpha, because they're all the same size. And then the zygote that results does not divide by mitosis. Instead, the zygote immediately goes through meiosis and forms 4 haploid individuals. So this is completely the opposite. So in this case, the only diploid cell is the zygote. Now, plants and some primitive plants, well, even some advanced plants, and algae have a mix of these 2 life cycles. They have a diploid version and they have a haploid version. Mosses and ferns have this. So the green leafy part of the fern or the moss that you see, that is the diploid individual. And then haploid cells form. So if you've ever looked at the back of a fern leaf and you see those little brown spore-producing circles on the underside of the leaf, those are the cells that are produced by meiosis. They are haploid, but they're not gametes. Instead, they drop into the soil and form a little haploid leaf. the little haploid individual, and then this little haploid individual then produces the gametes. And fertilization occurs, and then the zygote forms the adult. This is an alternation of generations in these. So although the human one is the one that we're most familiar with, which is like all other animals, and that is that the zygote is diploid, divides by mitosis, and the only haploid cells are the gametes. And the gametes can't do anything except go through fertilization. And if they don't, then they die. So for mammals, we have X and Y chromosomes for sex determination, and you've probably heard of that before. But that's not how all animals do it. Birds have W and Z chromosomes, and for birds, it's the female that has the two different chromosomes. Males have two of the same chromosomes, and the females have the W and the Z chromosomes. Reptiles don't have sex chromosomes. Which sex they're going to develop into is determined using environmental cues during development. And there's other organisms that don't even have gender at all, and they don't have two different size gametes. So the definition of male and female is male is the gender that produces the small gamete, female is the gender that produces the big gamete. So if there's if the gametes are all the same size, then it's not really gender as we understand it. So here's a nice scanning electron micrograph of the X and the Y chromosome. You can see that the Y chromosome is much smaller. And some people think that we call them X and Y because of the shape. But that's, I mean, all the chromosomes are sort of X-shaped. So that's not why. So what happens when it's not the case? As I said, for the vast majority of human beings, if you have XX chromosomes, you're going to be female. If you're XY, you're going to be male, but not always. Like everything in biology, there's always exceptions to everything. And the two exceptions to that we would call intersex individuals, and a different category is transgender individuals. So these are completely separate things. So intersex means that a person's physical measurable characteristics, their phenotype, they have one or more of each gender. So there are certain physical characteristics like what your chromosomes actually are. Are you XX or XY? Do you have ovaries or testes? Do you produce eggs or sperm? Do you have a uterus or other internal organs? What is the external genitalia like? Is there a vulva? Is there a penis? Those are external genitalia. And what are the hormone levels? What do you have hormone levels that are typical for male or are more typical for female? Now, sometimes intersex individuals are identified at birth because normally the baby's born, the first thing the doctor does, or the midwife, is say, oh, it's a girl, it's a boy. But sometimes at birth, they have ambiguous external genitalia, and which sex they are is not obvious. Sometimes intersex individuals are not identified until later in life because they don't go through puberty or other reasons. Sometimes external genitalia looks like one gender, but actually they have internal organs of the other sex. So, but it's a very small percentage of the population. So intersex specifically means people who have measurable characteristics, either their actual physical genitalia, external genitalia, internal organs, chromosomes, something, hormone levels, something we can see or measure that they have a mix of the two sexes. And they are far less than 0.1%. So 0.018%, that's a very small percentage, but it's important and significant. And the way that intersex individuals are being treated as children has changed significantly, luckily for the better. Surgeons used to routinely do surgery on intersex individuals as infants, assign them to female generally. and then never tell them they were intersex. And that turned out, now we know that that was not the right thing to do. And generally now intersex individuals are allowed to, because there's no reason for immediate sex assignment or surgery. They are allowed to grow and develop and then determine for themselves what they want to be, which seems like a much better way of doing things to me. Transgender is completely different. So transgender individuals, that means that their physical characteristics are all of one sex. So they are XX with ovaries and female external genitalia and hormone levels. They're not intersex individuals, but they don't feel like they are the right gender. So that's also less than 1% of the population. And we don't have any way to measure this at this point. It's all individual self-reporting. So the percentage could go up as it becomes easier and less risky for individuals to identify themselves as transgender. So right now it's less than 1% of the population, but 20 or 30 years from now it could be more than 1% of the population. So, but this is something that is not physically measurable. So very different things. There has been some controversy with the Olympics with intersex individuals. In the 2016 Olympics, a woman named Caster Semenya won the gold medal in the women's 800 meters. She is an intersex individual, so she was identified as female at birth, and a doctor also identified her as being intersex. Now, Caster Semenya also says that she identifies as female, and she considers herself to be a woman. But she is also an intersex individual. Intersex, remember, is something that we can measure. We can measure things on intersex individuals. It's not just a issue of self-reporting like it is with at this time with transgender individuals. So in that same Olympics, the silver and the bronze medals in the women's competition were also won by intersex individuals. So the gold, silver, and the bronze in the 800 meters were all won by individuals who were intersex, who all three of them have XY chromosomes, have high testosterone levels. And when we say high, we don't mean testosterone levels that XX woman might have if she has PCOS or some other medical condition. No, we mean high testosterone, they have hormone levels that you would expect to find in a normal man. So Caster Semenya, in fact, has XY chromosomes. She has testes, not ovaries. Her testes are undescended. They are in her abdominal cavity. She has no uterus, and she has the testosterone levels of a normal male. So the IAAF committee, the International Association of Athletic Federations, I think is what that stands for, before the next Olympics, which now has been postponed because of COVID-19, they said they were going to have to decide what they were going to do about this, because women's sports is a protected category. Anybody can compete in men's sports in the Olympics. Anybody could say, I'm going to compete in men's track. It's not a protected category. Women's sports have traditionally been a protected category, and there have been many, many, many attempts in the past to try to protect that category by keeping male athletes out of women's sports, because it's been recognized that having higher testosterone levels is an advantage that a woman who's not supplementing with steroids doesn't have. So what they determined from talking to experts was that the advantage specifically is having XY chromosomes. So they said any individual who has an X and a Y, if you're an athlete and you have a Y chromosome, the only way you can compete in women's sports is if you do something to lower your testosterone level. Now, any XY individual who identifies as a woman, like Caster Semenya, she could compete in the men's category without altering herself in any way. So that's the new rule. They're not going to test testosterone levels in women. It's not an issue of testosterone level for anybody unless you have a Y chromosome. So if you're a woman, the rule only applies to women with a Y chromosome. So women with a Y chromosome have to show that their testosterone level is within the range for women. And they're actually allowing a pretty high range for this. It's not a small deviation from normal, from your average XX woman. They're actually allowing a pretty high level. So probably Caster Semenya is not going to compete in the next Olympics because she indicated that she was not going to be willing to take drugs to lower her testosterone level. But that's the rule. And people have vastly misunderstood this ruling as applying to all women competing in the Olympics, and it absolutely does not. It only applies to women who have a Y chromosome because that's what they determined was the advantage. That if you have a Y chromosome, it will very likely cause very high testosterone levels. So that was that issue and how it relates to our topic. So specifically for intersex individuals. So before meiosis, before a cell is going to go through meiosis, we have to have pairs of chromosomes. If a cell is haploid, it cannot go through meiosis. So here we have an example cell that has 3 pairs of chromosomes, so of 3 different sizes. So one large pair, medium-sized pair, and a little bitty pair of chromosomes. And so the N number for this species would be 3. And there are different colors here to represent the fact that one haploid set came from the one parent and one haploid set came from the other parent. And on each chromosome, there are 2 identical sister chromatids, so that tells us that DNA synthesis has already occurred during interphase, and this cell is now ready to go through meiosis. So for meiosis, we're going to have 2 rounds of divisions. We're going to get 4 daughter cells, and the chromosome number is going to be reduced by half. And then that way when fertilization occurs, or syngamy, if it's a species that doesn't have a large and a small gamete, we wouldn't call it fertilization, the chromosome number, the diploid number, is going to be restored. So there are, besides cutting the chromosome number in half though, there are 2 processes that are going to occur during meiosis that are going to increase genetic diversity. Remember in evolution we talked about variation being important. Meiosis and sexual reproduction increase variation in a species because all the offspring of 2 parents are going to be genetically unique, and meiosis helps that to occur. So we'll talk about 2 processes that occur that increase variation. One is crossing over. Crossing over is where the 2 homologous chromosomes swap some of their DNA. So we end up with chromatids that are unique. And also independent assortment. So the chromosomes that came from the maternal lineage originally and the paternal lineage originally, they don't necessarily stay together when this cell is going to divide. And every time meiosis occurs, the daughter cells are going to get a different assortment of chromosomes that had a maternal or a paternal origin. So here we have our pair of homologous chromosomes, and the first weird thing that happens in meiosis is crossing over. So synapsis is the pair of homologous chromosomes coming together. Crossing over is the double strand DNA is actually going to be broken and reattached. So we're going to end up, after crossing over, and this occurs in the first prophase of meiosis. We call the divisions in meiosis by Roman numerals, so this is a Roman numeral 1. So the first, everything associated with the first division has a Roman numeral 1, and the second division has a Roman numeral 2. So we have prophase, metaphase, anaphase, telophase 1, and then we have prophase, metaphase, anaphase, telophase 2. So crossing over occurs before that first division, and you can see what the result is here, is that we end up with chromatids that are different. So we have an unaltered chromatid here, and then we have two chromatids that have a mixture of DNA from the two different parents. And this is going to increase genetic variation in the offspring. The second thing that increases genetic variation is what we call independent assortment. Independent assortment is how those pairs of chromosomes line up during the first metaphase. So it could be that all of the paternal ones are going to go one way and all the maternal ones are going to go the other way, but it could be any combination of those as well. So they line up randomly, the pairs of homologous chromosomes, and when they separate to reduce the chromosome number by half, which way they go is random.